blood, and this is called the absorption state. All right, so the first couple of hours or so after digestion has occurred, and we have higher glucose, higher fatty acids, higher amino acids in the blood, is the absorptive state. And primarily, what organ is going to be involved in reducing the levels of amino acids and simple sugars? Of all the organs we've talked about. What's the largest gland within the body?
The liver.
The liver. Okay. So along the sinusoids, through the fenestra of the sinusoids, and across the space of the same, and into the liver. Remember all the glycogen, those dark pumpy things I showed you? The liver is going to store simple sugars as glycogen. We're going to take the amino acids and, along with other cells, but primarily the liver, is going to take the amino acids and make proteins. And then the lipids, if they're excessive, will move into adipocytes if they're not taken up by additional cells. So, of all of those three different food types, glucose is the main one that our body is set up to use for energy. Okay? It can use fatty acids and it can use amino acids, but glucose is the main source. So I'm not going to go through all the steps. Like I said, if you haven't done this in micro, you will. Okay? But these are the three main components that occur as we get energy from the carbons, breaking the carbon-carbon bonds. Where do we get our energy from? What part of the molecule of glucose or fructose do we get our energy from? Electron. Okay? So the three steps are glycolysis. The Krebs cycle is a little older fashioned name, so citric acid cycle. And electron transport. Alrighty? What I'm writing on the board is what I want you to know about this, okay? There might be more detailed questions on the quiz, but that's an extra credit quiz. So, but as far as the exam, the level of detail I'm going through tonight will be. Glycolysis takes place in the cytoplasm. It is regulated by enzymes. Okay? So, back to Bio 430. The enzymes that regulate the steps of glycolysis would be synthesized using the ribosomal ER and Golgi or without that. Going back to Bio 430 again, and what we talked about with the lipid activity, we use ribosomes and messenger RNA attached to the ER, inserting the new protein into the lumen of the endoplasmic reticulum and being modified by the Golgi when that protein has a destination, a membrane-bound destination. So if it's going to the lysosome, or if it's going to the mitochondria, if it's being excreted from the cell like the chylomicron, okay? If it doesn't have a membrane-bound destination, it's staying within the cytoplasm of the cell, then it's using a free ribosomes, polyribosomes. It's not involving the ER or the Golgi at all. So these enzymes are going to be synthesized via polyribosomes or free ribosomes. So polyribosomes are seen as little clusters of ribosomes like that, so they look similar to the glycogen I showed you in the liver, but much smaller, okay? This is considered anaerobic because it will take place even if oxygen is not available for a long period of time. But it's not very efficient because it only nets 2 molecules of ATP. It actually makes 4, but we have to get 2 to prime the pump and get it going. So it nets us 2. All right? We don't make any carbon dioxide. We're not releasing any carbons in glycolysis. Between glycolysis and the citric acid cycle, there is an intermediate step in which carbon dioxide is released. Okay? 2 of them. So this is where the carbon dioxide comes from that we are exhaling in our respiratory cycle. So what we start out with the citric acid cycle is a 2-carbon molecule. We have 2 of those. And we add it to a 4-carbon molecule, which ends up being our citric acid. And it goes through a whole series of changes. In other words, we made different amino acids from that. So citric acid to succinyl, different components. And along the way, CO2 is released. By the time we get done with our entire glucose, 4 molecules of CO2 have been released. So we're done with all of our carbon, okay? Now, in addition to carbon and oxygen, our glucose and fructose has hydrogens, okay? So the hydrogens are sent to the mitochondria component. Both of these, citric acid cycle and electron transport, are taking place in the mitochondria. So the hydrogens that are captured on either glycolysis or the citric acid cycle, and the citric acid cycle enzymes are taking place inside here, okay? And so the hydrogens are where we get the electrons from which we get our energy. So they use coenzymes, so we have these molecules NAD and FAD. So that's how we bring the hydrogens, and we get rid of the carbons and the oxygens, so all we have left from our glucose or fructose is hydrogen. The hydrogens are then split. We have a hydrogen ion, and we have an electron. Hydrogen only has 1 proton, okay, and 1 electron. And so the hydrogens are pumped into the outer matrix. Where does the energy come from to pump the hydrogen ions up here? It comes from the release of energy as we move the electrons from one molecule to another. That's electron transport. Okay? So electron transport, we have enzymes embedded in this inner membrane. And as the electron, one wants it more than the other one. And it's like taking a bucket of water and filling it with water, and that next bucket doesn't need as much water, so there's water that's left over. We have time. I'm going to tell you a story. Apologize to the women in the audience because it's not doesn't treat women in a very good light. Okay? So there's this woman who's alone, and she goes to a bar, and she meets up. This is a heterosexual story. She meets up with a guy, and they're really taken with each other. She goes home with him. And as they drive to his place, they're going in a Jaguar, which is what I would buy if I had $2 million and didn't care about the upkeep. And they park in a garage that has 12 other cars. And then they get in a helicopter and they fly across the water to the island where the house actually is. And the next morning she wakes up, in her glass of orange juice are 5 5-carat diamonds. She's like, wow, this is a nice guy, and he has money. I told you it doesn't put her in a very good light. But after 3 or 4 days, she's tired of him. And he's traveling a lot, and even though he gives her 25 platinum cars and the helicopter at her disposal, she didn't go home with him to spend this money. She didn't think, she realized she wanted companionship. So she finds herself back at the bar. She finds another guy. He's paying her even more attention. She goes home with him. Told you, it doesn't put her in a very good light. Well, he only has 5 cars in his garage and only 2 5-carat diamonds in her glass of orange juice. And she doesn't realize that he's making just as much money as the other guy, Will ignore the fact that she has her own income, just making some parity there. And he's taking that money that he doesn't need to spend on her, and he's putting it into investments. But she still stays with him because affinity, she likes him better. So that extra money he's not spending on her, he's saving, pumping hydrogen ions into the outer matrix. But even he too is too busy for what she wants, and she goes back to the bar. And this keeps happening over and over, and the guys she goes home are just as rich, but she likes them more and more and more, and they have to spend less and less money to keep her. And finally at the end, hydrogen plus half of an oxygen plus our electron all come together to form our final product, which is water. And guess where she finds herself living? Completely hysterically happy. A little shack on the beach by the side of the Pacific Ocean, next to the water. All right, so the electron starts at a high level of energy, very tightly attached to its NAD, but FAD, quote unquote, wants it more. And so it moves to the FAD. And there's enough energy left over between how much NAD has to spend to hold on to the hydrogen, how much FAD has to spend to hold on to the hydrogen. That energy is enough to pump hydrogen ions into the outer matrix. And that keeps going until we've moved hydrogen ions against their gradient into the outer matrix. Well, the whole purpose of this is to store energy as ATP, right? So imagine instead of pumping hydrogen, we're moving water to the top of a water tower, a little bit of energy at a time. And now we're going to let that water all run down a pipe, and it's going to spin a turbine. And that creates electricity, which we store. Where's the turbine in this event? So we're looking, in other words, if we release all of this energy at the same time, it would be too hot for the cell, okay? So we're releasing it a little bit at a time as we move that electron along the molecules of the electron, the membrane. So here's what we're doing. We're moving electrons from these molecules one to the next to the next. And the energy in between is moving hydrogen ions to the outer matrix. And finally, when it's gone to its lowest level of energy, all right, it recombines with the hydrogen ions and the oxygen. This is literally where we use the oxygen that we breathe, is in this last reaction here. And now, all the hydrogen ions that are in the outer matrix come down through this channel of this molecule known as an ATP synthase. And it literally spins. It's a little motor inside your mitochondria. So I had another image of it. And as it spins, it creates energy that takes that third phosphate and adds it to ADP. So ATP synthase is this protein molecule that takes the energy of the hydrogen ions coming down through that channel to add that third high energy phosphate group. So at the end of all of this process, we end up with ATP, water, and heat. Okay? Now each of these would have its own end product, okay? But as a result of all of this, we end up with those components. Does it have to be glucose that we start at? Actually, glucose gets modified and becomes fructose, so it's a little easier to use fructose. It doesn't. We can use, when this is split, glucose and fructose have 6 carbons. When those are first split at the end of glycolysis, we end up with 2 3-carbon groups. Remember our glycerols that we talked about? This is forms a molecule for free known as glyceraldehyde. So glycerol from fats from our triglycerides can come in here and be inserted, all right? Many of the products of our citric acid cycle are amino acids. So amino acids can come into the citric acid cycle, and be used for energy. So, I thought I had another term. All right, I'm going to write those terms on the board. So there are a few words that sound very similar that I want you to know of as definition, okay? So, any questions on this? I know I went through it really fast, but let me reiterate. 3 stages of getting energy from primarily carbohydrates, all right? The first does not require oxygen. The last 2 will not occur without it. Glycolysis takes place in the cytoplasm. It makes 2 molecules of ATP. By the way, the other 2 make a total of 36. So it's a big difference between what we can do with oxygen and without. Citric acid cycle, electron transport, takes place in the mitochondria. So they're going to use enzymes that have come through the ribosomal ER and the Golgi. Okay? Our byproducts, in addition to the molecules we form along the way, this we get rid of the carbons as part of our sugar group as carbon dioxide. And we're left with just the hydrogens. Those are converted in the mitochondria and split to form hydrogen ions and electrons. And the energy is actually in the electron. It starts at a high energy level. As we bring it to lower energy levels, energy is released. Because each subsequent molecule that I have up there doesn't need as much energy to hold on to the electron. So that energy is available to be used to move the hydrogen up its gradient into the outer matrix. All right? One of these molecules is cytochrome oxidase. Where have we heard that before? Histotoxic hypoxia. The mitochondria don't work. Oxygen can't be used with poisoning due to potassium cyanide. So it just freezes that step. And oxygen is never used in its final step. So it stays in the capillary and the oxygen returns to the lungs without being used. Doesn't mean the cells don't need it, they just can't use it without that enzyme. Okay? All right. So your lecture notes have a lot more detail with a summary at the bottom of each of these events that if I had lectured on it would be what I wanted you to know. So when you're taking the extra credit quiz, that's a good place to look. Okay, so you'll see a little box or a list of points at the end of each of these components. All right.
Can you explain really quickly where the NADH2?
So these are called coenzymes, which means they will function as an enzyme to strain the relationship of the chemicals, but they function as coenzymes by bringing the substrate to the region. So when hydrogens are released, as we go through these different chemical changes releasing the carbon dioxide, hydrogens are also released. They're captured by these molecules, okay? And brought either to the cristae or brought from the cytoplasm to the mitochondria, depending on where their source is. Okay? All right. So again, absorptive state is right after you eat, when blood levels of the digested food are high, and post-absorptive is the state where those have already been taken up into the cells, so you haven't eaten it for a few hours, and your glucose levels are low, and fatty acid levels are low, and amino acid levels are low as well. All right, so we have glycolysis. So glycolysis is the breakdown. This is This is catabolism. So metabolism is, I think of catastrophe. So it's the breaking down part of metabolism. Anabolism is the making together. So glycolysis is the catabolism of glucose, primarily, or any other food stuff, to form this is the end product of glycolysis before we go into our citric acid cycle. This is our 3-carbon molecule, okay? If we don't use glucose for this process, and we use amino acids or we use fatty acids. Remember the fatty acid was a long chain of carbons with a fatty acid at the group. These can be broken into 2 carbon molecules, which is what goes into our citric acid cycle. So fatty acids can be broken down into these 2 carbon components and go directly into the citric acid cycle. So that's an example of gluconeogenesis, when we use something other than carbohydrates to get energy, okay? So this is the use of fatty acids, amino acids, or lipids such as glycerol, and glycolysis or the citric acid cycle, okay? Unfortunately, they all start with a G. So glycogenesis, what's our storage form of carbohydrates? Starch, and that's what glycogen is, all right? So I showed you those starch molecules in the electromicrograph. So glycogenesis takes place in all of our cells, but the major place is the liver and the hepatocytes. So this is anabolism, building up. So this is taking sugars, simple sugars, and forming polysaccharides. Okay. Now, if we don't have enough glucose in our blood, then the liver is going to break down glycogen under the influence of a hormone from the pancreas to increase, this is homeostasis, so we don't have really deep, low levels of glucose and really high levels, we kind of moderate it out. So this is the formation of polysaccharides, and its opposing term is glycogenolysis. So breaking down, this is making glycogen, this is breaking down glycogen, okay? So this would be the catabolism, this is the reverse. So within the cell, taking polysaccharides and returning them to simple sugars, to monosaccharides. So which of these would be the absorptive state? Glycogenesis or glycogenolysis? Which of these are we going to do right after you finish eating when blood glucose levels are high? Glycogenesis. And then the post-absorptive state, so it's been 6 to 8 to 12 or maybe just 30 minutes and you're hungry because you ate high carb thing, then we would go to glycogenolysis, okay? So, just as a review, before we get to the hormones, just looking at this pair, insulin and glucagon are the primary, glucagon specifically for the liver. All right, and then one more thing before we look at the hormones. And we're going to be done in 15 minutes, whether you think so or not. My mouth stops talking at 7:31. That would be 17 minutes. All right. What's the difference between a calorie? May I erase the terms? You guys have that? So a little C calorie is a measurement of energy release. And we measure the amount of energy as heat, okay? So it's specifically done in what are known as British thermal units, and I'm not versed enough in physics to compare that to non-British thermal units. But when we define a little C calorie, all right, it is the amount of energy And heat is a form of energy. All right, so you may have heard of a bomb calorimeter. They put food in a thing and not blow it up, but release energy from it by heating it. So the amount of energy needed or released to increase the temperature of 1 mil of water, that's a cubic centimeter, by 1 degree Celsius. Okay? A kilocalorie is the same as when you look at nutrient information on packaged food, it's the same as a large C. Okay? So little c versus large C. Kilo is a magnification of what? 1,000. Okay? So calorie or kilocalorie is equal to the amount of energy which will raise the temperature so can I say the temperature of 1 mil of water 1,000 degrees? Does that work? What temperature does water boil at in this system? 100. So should I raise it to 1,000? It'll never go higher than 100. So this definition doesn't work for those of you that, I'm sorry, wrote it down. I can't do it that way because it'll never go higher than 100. So to raise 1,000 mils of water, which is a liter, 1 degree. Okay? I suppose you could say 500 mils, 2 degrees, but we can't raise it 1,000 degrees. It won't go to that temperature. Water won't go higher than 100. All right? So kilocalorie, basically a slice of bread has 100 kilocalories, depending on how thick it is and big and all that kind of stuff, and how much peanut butter and everything you put on it. But just a slice of bread is about 90 calories. Okay? So we're talking about needed to raise 900 mils of water, 1 degree, when we say 90 calories. Okay? All right. Basal metabolic rate. BMR, or your basal metabolic rate, is the amount of energy you need to live. Not to think, not to walk, not to drive your car, just the bare minimum of your cells to stay alive. Okay? For most individuals, that's given as 1,200 calories. A lot of people that will gain weight on 1,200 calories. Okay? So it's going to vary based on your own. Some people eat 2,000 calories and still lose weight. So bare minimum with plus or minus 400. So, but that's what the definition. Anything else you do above your basic cell activity to stay alive is going to have to be added on to that. Okay? Okay, difference between malnutrition and malnourishment. If you are taking in your basal metabolic rate plus the amount of calories you need to run the 2 miles you do every morning and do your work and everything else, all right, you are not malnourished. You have enough calories. But if you have, let's say 100% of those calories come from carbs, and you don't have enough vitamins, then you are malnutrition, okay? So malnutrition has to do with the imbalance of the food stuff, vitamins and minerals and fats and carbs and proteins. You might have the adequate calories, that's what nourishment is, all right? So malnourishment is too many or too little calories. Malnutrition is too many or too little balance of the different minerals and vitamins and carbs and proteins and lipids that your body needs. I've got 12 minutes. All right, so we haven't even started the hormones, and I'll tell you why this is. It's okay, I don't mind. So we have a plane flying over the desert. The pilot of that plane is a 90-year-old woman. I move her around every time. She's got her senses, though, okay? So we have a 90-year-old woman. She has her 30-year-old granddaughter, who's about 150 pounds. She's about 110. The woman just gave birth, and she has her 6-month-old son with her. Okay? The 30-year-old woman has a twin. And this woman is about 380 pounds. Okay? The 9-year-old woman didn't realize she had cataracts, and she crashes the plane, flying into the setting sun. And which of these? They have some water on board, but not much, and no food. Which of them has the best chance of surviving? And they're not cannibals.
The 30-year-old? The obese one.
The obese woman. Couple of things going on here. The 6-month-old son and the 90-year-old woman their hypothalamus and their ability to maintain homeostasis through temperature control are not mature or are not working well. Okay? So their ADH secretions, all of that, are not going to be off. 30-year-old woman has a lot of protein. Even though she just had her son, she's training for a marathon. When we break down protein for fuel, we only get about 4 kilocalories per gram. Whereas when we break down fat, we get 9. Plus, protein has what element in it that fat does not? We just finished a unit. Urea, ammonia, creatinine. It has nitrogenous waste products that are going to require additional water to flush out. So if she's using her protein for fuel, she's going to need way more water to flush out those byproducts than the woman who has a lot of adipose tissue. Okay? So for a couple of different reasons, she has the best chance of survival. Okay? Which gives you permission to go home and eat Christmas cookies. If they're covered with cream cheese frosting, that is. Okay. This is a lot of information. How many minutes do I have left? 5? 10? Okay, we'll go to the end. This is what you need to know. Let me explain why. Okay? So these are the hormones that affect metabolism and the use of energy. Insulin functions, functions to help certain cells, not all cells need it, but insulin functions to help certain cells by increasing the glucose transporters that are inserted into the cell membrane, similar to how ADH causes aquaporins to be inserted. Insulin causes glucose transporters to be added to cells, and so more glucose is taken up. All right. It also breaks down proteins for energy sources and lipids. Not breaks down, but uses those for components. And so the levels for amino acids and fatty acids decrease as well because they're utilizing them in that gluconeogenesis. Glucagon really only affects glucose, all right, by causing it to be released from the glycogen formation. So glucagon is going to do the glycogenolysis. So you're going to see an increase in the levels of glucose. Now from there, I want to switch to growth hormone. I used to have students do case studies. And I had a case study, his name was Tyrone. He was a bodybuilder, and he was using steroids. And after 2 years, he ended up with diabetes. Because the growth hormone anabolic steroids that he was using caused the body to increase growth, so it was decreasing the level of amino acids in his blood because it was making proteins, all right? It was breaking down fats, which is a good thing if you're wanting to lose weight. So it was increasing the release of fatty acids because it was breaking down triglycerides. But it was using those instead of fuel, so the glucose levels stayed high because they were preferentially, it was a glucose sparer, okay? So instead of using glucose for fuel, it was using fats. Great way to lose weight, all right? Build muscle, sounds like a perfect weight loss type of drug, but it wasn't allowing the cells to use the glucose. So the glucose levels in the blood stayed high, which caused a wearing out of the pancreas. So the pancreas was overstimulated to release insulin because of the chronically high levels of glucose. Does that make sense? Okay. So in the short term it looks great, but it ends up causing an overproduction of insulin. Okay. There can be either just the cells no longer make it anymore or a loss of sensitivity when you have high levels of that. All right. Cortisol. Cortisol is also a glucose sparer. So glucose levels are increased. Cortisol is a hormone from the adrenal glands. All right, we talked about aldosterone, testosterone. There's 3 different levels. It's increased in inflammation, or rather it's increased in stress, and it reduces healing. It's a glucose bearer, and it breaks down muscle and fat, okay? So the reason we have an increase in amino acids with cortisol is because it's increasing the catabolism of proteins. And I don't know if I mentioned this when we were talking about cortisol back in reproductive, but I think I did, where the woman that was taking care of my babies mentioned her husband, not her husband, her father, had dropped a tool in his garage, and when he bent down to get it, he couldn't stand back up again, and he had elevated levels of cortisol, okay? So the weakness came from the breakdown of the proteins in his lower limbs that would then, over here, that would then increase the amino acids, okay? Fats are broken down, so it increases fatty acids. Glucose is not used, so it stays elevated, okay, instead of being taken from the blood and used. Thyroid hormone is a little bit more vague, all right? Thyroid hormone increases the metabolic activity of the body. And so to provide for that metabolic activity, we need to make more ATP, and therefore glucose is released from cells and fatty acids are released. It's more of a, not as tight a link to what those numbers are, but that's the reason why, okay? So in going back, you don't have to worry about the epinephrine. This is the cortisol, okay? So I have it more specific as to what's going on with each of these, but that's the reason why, okay? So when this is a written fill-in portion like I normally have on exams, I would have a table with, like you would see, let me go back to the table. These components on it, what organ makes insulin, what organ makes glucagon, what organ makes cortisol, and then one or two of a couple of these is an increase or decrease it type of a thing, all right? So I have, with 50 questions, I can't have as many as when I had 80 plus 20. So, but I will have a couple of these on there, but as a multiple choice type of question, all right? So the effect of cortisol is to increase or decrease amino acids.